Hello, and welcome to our team's presentation about the drug Ivacaftor. These are some presentation housekeeping notes, and you can take a look at these at your leisure. Our team is comprised of 4 members with various backgrounds, and before we get started, we would like to thank our team leader, Anna DePaula, for establishing the team and sharing her knowledge with us. Just so you will know, Anna is located in Pittsburgh, Pennsylvania. Rajesh is in India. Emmanuel is in Nigeria. And Amelia, which is me, lives in Cary, North Carolina. We've provided many biographies of our team members, so again, you may read these at your leisure, and we're going to get on with the presentation. Ivacaftor truly is a phenomenal leap forward in the treatment of the genetic disease of cystic fibrosis. So to gain a better understanding of just how exciting this therapy is, we provided a bit of an overview of the disease as well as the drug history. It is defects in the CFTR protein affecting the transport of chloride and other ions across cells that cause cystic fibrosis. This leads to an accumulation of thick and sticky mucus in the lungs of CF patients. And Ivacaftor is the new drug therapy that treats certain CF patients by significantly improving their breathing. It works directly in the lungs, which is the great thing about it. Cystic fibrosis is the most common lethal autosomic recessive disease among Caucasians. It affects about 1 in 2,500 births. And CF is characterized by chronic respiratory infections, which is the main cause of death. There's pancreatic dysfunction, elevated sweat electrolytes, and also male infertility. Cystic fibrosis affects the lungs and digestive systems of about 30,000 children and adults in the United States and 70,000 worldwide. Cystic fibrosis is thought to be almost as old as humans themselves. Interestingly, CF patients will not contract cholera. The thinking is that somewhere in our genetic development and based on Darwin's survival of the fittest, at some point CF won out, if you will, over cholera, and the CF gene mutated and began to be passed along. So in the 1950s, few children with cystic fibrosis lived to attend elementary school. And today, advances in research and medical treatments have further enhanced and extended the life for these children and many adults with CF. Also, many people with the disease can now expect to live into their 30s, 40s, and beyond. The market. Ivacaftor was researched and commercialized by Vertex Pharmaceuticals. Vertex is located in Boston, Massachusetts. As you can see in the timeline, Vertex acquired Aurora Biosciences in 2001. It was Aurora Biosciences who had the chemical compound and the novel assay equipment that was especially well suited for CF research. And we're going to talk a little bit more about that later. And also of note, Aurora Biosciences, now Vertex, is located in San Diego. So the majority of the research took place in San Diego for the drug Ivacaftor. In the 1980s, when the CFTR gene was discovered, research really began to accelerate around CF. And additionally, treatments for the symptoms of CF had steadily improved. There were better antibiotics, better breathing treatments, and the like. So what's so wonderful about Ivacaftor? It is the first drug that actually treats the underlying disease and not just the symptoms. So from the time the CFTR gene was discovered until the drug went on the market was about 23 years. So in the scheme of things, this was pretty fast. The market opportunity for Ivacaftor is excellent, despite its hefty price tag, which is about $234,000 a year for a prescription. But we also need to remember that this drug will have to be taken for life. For those of you in the U.S., you may be familiar with Jim Cramer, who has a TV show about stocks and the stock market. He's an excitable kind of guy who yells out the word booyah when he thinks that a stock is a good thing. And it's Kramer's opinion that Vertex will someday dominate the cystic fibrosis market and generate annual sales close to $10 billion. And they will do this with little or no competition. Maybe this is a good stock tip for those of you investors out there. As for benchmarking, this is our team's definition of benchmarking analysis. It relates to stochastic modeling for all of you math majors who understand things such as that. So this is how we applied this to Ivacaftor and regarding the decision of abandonment. So given the fact this drug works, there was obviously no decision of abandonment. When told that most medicines worked or would never make it to patients, Ivacaftor, which is now marketed as Kalydeco, fits with Vertex's founding philosophy. And Vertex Pharma was founded in 1989 by Dr. Joshua Boger, and he believed that there was an idea that no challenge should ever be too great. So the burden of disease for cystic fibrosis is huge, as you can see by this graphic. The patients are usually underweight and have a hard time gaining weight. They're almost constantly battling lung infections. So there's a lot of medications and a lot of hospital visits for some of these patients. Clinical development. And so we're going to begin with preclinical. This slide shows a graphic that depicts the cell and the CFTR gene that gives rise to CF. And this is the cystic fibrosis transmembrane conductance regulator. That's what CFTR stands for. So in order to have a fully functional CFTR ion channels, they are embedded within the membrane, and three basic processes need to be completed at a cellular level. First, the CFTR protein must be correctly synthesized. Then the synthesized CFTR protein must be properly processed into a mature channel. And thirdly, the CFTR protein channels at the cell membrane must function properly. So for the wild type CFTR protein, the newly synthesized protein traffics to the cell surface of the epithelial cells and functions mainly as a chloride selective ion channel. So alternately, the mutant protein, and here we're showing the one particular mutation which most people with CF have. It contains a deletion that results in the loss of phenylalanine at the FO8, which is the gene number designation. So this protein is essentially misfolded, and it gets retained inside of the cell, and it never reaches the cell's surface to work properly. So this makes the body produce unusually thick, sticky mucus that affects the lung and the pancreas. These are images of actual CFTR cells, and the top image is of a normal CFTR cell, and the green bar is the channel through which the ion passes. Alternately, the delta F image shows the mutated cell and hole in the channel. And there's the delta F, the abnormal gene. This figure illustrates current and future therapies and the pathogenesis of CF lung disease. Even though the median predicted survival age has increased over the years, most of the therapies are aimed at the latter end of the CF disease. However, the ultimate goal is finding a cure for CF. So we need to identify targets and small molecules that will address this cascade earlier. This slide demonstrates the pathophysiology of CF, from the defective gene all the way to the end stage of lung disease that was already previously shown. So in the lung, the proper water balance is important to maintain the proper height of the airway surface liquid, otherwise known as ASL. And it's the layer on the inside of the epithelial cell membrane. And so the cilia, if you remember from your basic biology, the cilia project from the surface of the epithelial cells into the airway surface liquid layer. So it's the fluidity of the normal ASL layer that enables the cilia to beat and clear that mucus that is trapped with particulates from the lung. So in cystic fibrosis patients, the ASL is reduced and the normal ciliary clearance is defective, and it results in the mucus buildup. The cilia can't beat the way it needs to beat to get all that bad stuff out of there. And this is the process of the CF lung disease. So the CFTR modulators are drugs, which in this case is the Ivacaftor, that targets specific defects caused by the mutations in the CFTR gene. So in keeping with Vertex's founding philosophy, the idea that no need is too great, the Cystic Fibrosis Foundation began a collaboration, and they had been looking for pharmaceutical companies that would work with them on the research, and they found Vertex. So this is a timeline of the events leading to the commercialization of Ivacaftor. Initially, the CFFT gave Vertex $73 million to start their research. And here's what's one of the interesting things about Ivacaftor. The identification of the novel CFTR modulator was achieved by performing high-throughput screenings at Aurora Biosciences, which is obviously now Vertex. And it was their technology that allowed them to go through large chemical libraries using cell-based fluorescent assays in rapid amounts of time. As you well know, assays can take a very long time. But this equipment that was then owned by Aurora did this really, really fast. So this particular assay has to measure the main function of the CFTR protein, and in other words, the transmembrane transport of chloride and other small ions have to be measured. So the CFTR function may be determined directly with electrophysiological techniques. However, these methods are really time-consuming and expensive. So high throughput screenings are better performed using fluorescence-based techniques, which was the case for the CFTR, for the CFTR. So from this screen, multiple rounds of medicinal chemistry optimization were undertaken to improve the lead compound's potency and selectivity. And this is really where the shot in the dark occurred. So essentially what Ivacaftor does is that it keeps the channel open so that the ions can flow through as they would normally. And exactly where this compound binds and precisely how the compound works remains unknown. And obviously there are theories, but like we said earlier, this was literally a shot in the dark. And most therapies for patients with CF focus on managing the clinical consequences of the disease. But modulators like Ivacaftor is a therapeutic approach that targets the underlying protein defect that causes CF by modulating the CFTR protein function, which results in enhanced ion transport. Currently, more than 1,900 mutations can cause CF, and every week new ones are discovered. Now, these mutations are classified in different classes according to the mechanism by which they disrupt the synthesis, traffic, and function of the CFTR. So, CF patients with the G551D mutation can be treated with Ivacaftor. This particular mutation is when the misfolded protein comes to the top of the cell, and there is Ivacaftor waiting there to help hold the channel open. And this essentially makes the protein work as though it were normal. However, most patients with CF have the mutation that leaves the misfolded protein down in the cell where it degrades. So what Vertex is working on currently is a way to pull those defective proteins up to the top of the cell so that Ivacaftor can do its job. The G551D mutation is where a single nucleotide base pair is substitute for another at position 551 in the protein. So why is this important? Well, it's because CFTR needs ATP to work properly. And the G551 mutation is located within the ATP binding region of the CFTR protein. And so basically, ATP opens and closes the channel or door so that the chloride can get outside. So patients with this particular mutation have a defect in the opening of the channel, a process called channel gating. So chloride transport through G551D CFTR protein channels is extremely limited compared to normal CFTR channels. This is another visual of how Ivacaftor works. And again, it only works in patients with the G551D mutation, and in the U.S. that means about 2,000 people. However, as we mentioned in a previous slide, Vertex is working on another compound called VX809 that will work in about 30,000 patients. At least that's what they hope. And this all relates to the homozygous and heterozygous mutations, if you remember your Mendel. And so in short, the VX809 works in those with the homogeneous mutation of CF. That leaves approximately 20,000 patients with heterogeneous mutation, and that's the most difficult to treat. But Vertex believes that it can get to these 20,000 patients with second-generation correctors like VX661 and VX983. Of note here is that the compounds applied to the rodent cells, which are obviously cheaper and more available, demonstrate a hit. And then they're tested on the human bronchial epithelial cells. However, most of the animal models that are usually used in preclinical data are not good models for CF because these animals don't develop CF airway disease. Some researchers, however, are using pigs since pigs and humans share similar biological characteristics. And here's just additional information, sort of synopsized for the CFTR protein. So based on the in vitro findings we described earlier, a phase 2 study was initiated to evaluate the safety and efficacy of Ivacaftor in patients aged 18 years and older who had the G551D mutation on at least one CFTR allele. Again, that's remembering your Mendel. So the takeaway message is about the endpoint involving the FEV1-FVC ratio, also called Tifano-Pinelli index. It's the calculated ratio used in the diagnosis of obstructive and restrictive lung disease. This represents the proportion of a person's vital capacity that they are able to expire in the first second of expiration. In other words, like if you've ever had a pulmonary fit test and breathed into a spirometer, they measure how much air is coming out of your lungs and can tell how well your lungs are working. And this was the criteria they used ultimately for their endpoints. So Ivacaftor is taken daily in tablet form, and in their studies, the PK studies showed, or pharmacokinetic studies showed, that the half-life of the drug required dosing every 12 hours. When the IND was filed, the FDA granted fast track designation to Ivacaftor, and if you will remember in one of our lectures, the FDA was described as a bad boyfriend. Well, if your drug gets fast track designation, that bad boyfriend just got a little better. Moving on to early clinical development. You know, as we say in the United States, this is where the rubber meets the road. And so this is an overview of the phase 1 and phase 2 trials. It's worth noting that a secondary endpoint in phase 1 was a chloride measurement in sweat because they were looking for a reduction. And interestingly, in the healthy volunteers in the phase 1 study, those without CF, Ivacaftor made them sweat better. However, the best news was that there was no significant adverse events. So as we indicated earlier, better lung function was what they wanted, and these are some of the actual results from a phase 2A study that established Ivacaftor's efficacy. Moving on to late clinical development. Among the several phase 3 studies that were conducted, these are examples of 2 pivotal trials. There was the STRIVE study and the ENVISION study. All things being equal, these numbers represent significant improvements in patients. This is how the STRIVE study was designed, and as you can see, it met its endpoints. Same thing here with the ENVISION study. As you'll remember, the pharmacokinetics is what the body does to the drug, and this is, like from absorption to elimination and all things in between. So here is where Ivacaftor really accomplished what the researchers and clinicians set out to achieve. This is worth noting, and because of the safety and efficacy of Ivacaftor in patients younger than 6 years old has not been evaluated. This is because the lower age limit for eligibility in the phase 2 clinical trials had a criterion established that limited utility of traditional spirometric measurements in patients under the age of 6. A spirometer is a device for measuring timed expired and inspired volumes, and so hence it indicates how quickly and effectively the lungs can be emptied and filled. We mentioned this earlier. So due to patient cooperation that was required, and spirometry can only be used on children old enough to comprehend and follow the instructions. It's pretty detailed. That's the reason why this drug was not tested in children younger than 6. So these are some of the questions that came out of the phase 3 studies, and they're still currently being addressed. So apart from the really good improvement of lung function, CF patients also gained weight. And that's notable. CF patients have a really hard time gaining weight. Also, this is a good time to mention that Ivacaftor is best taken with fatty foods, like, for instance, similar to vitamins like A, D, E, and K. Ivacaftor is fat soluble. Regarding the pharmacoeconomics of Ivacaftor, obviously more emphasis is being placed on pharmacoeconomics these days. And this is the only example that we could that we could find involving Ivacaftor. So of note here is that approximately 11% of the Irish population are CF gene carriers, or cystic fibrosis gene carriers. And interestingly, the NCPE initially determined that Ivacaftor was not eligible for reimbursement, or rather they didn't recommend that it be paid for by their healthcare system. However, sometime between January and February of this year, the NCPE reversed that determination and made it available. So on to the regulatory strategy, and this is where Ivacaftor now becomes Kalydeco. While the clinical trials were going on for Ivacaftor, Vertex's regulatory affairs specialists were determining how to get Ivacaftor on the market the fastest way possible. And this is where they made sure that they crossed all their T's and dotted all their I's. And one of their strategies was to get FDA fast track designation for Ivacaftor. This was the same thing for the EMEA. Vertex wanted to get Kalydeco on the market as soon as possible. So part of Vertex's regulatory strategy involved looking at other selected rare disease product approvals and comparing the basis of approvals from a statistical standpoint. This is similar to what pharma does for every drug that goes on the market. This is the information from the package insert that had to be written for approval based on the regulatory strategy, ranging from how the studies were powered to compilation of standard operating procedures, SOPs, throughout the process. And as many of you may know, there are a lot of SOPs associated with bringing a drug to market. Again, this is more information from the package insert that has to do with the dosage form, the schedule, and the patient population. Intellectual property strategy. Well, after working on the compound and fine-tuning it for their purposes, Vertex filed its patent in June of 2005. Since then, they filed patents on 2 more compounds that they will use in cystic fibrosis research. Obviously, this is self-explanatory. There is no litigation concerning Kalydeco at this time. As for asset protection strategy, currently Ivacaftor doesn't really have any competition, and their patent has 12 years before it expires. So in terms of asset protection, they're pretty safe for a while and for the foreseeable future. The marketing strategy. Vertex had a built-in marketing mechanism, if you will, through the Cystic Fibrosis Foundation. They also had a blue ocean market. And a blue ocean market is a term that was coined a few years ago to describe how a product or a service creates demand in an uncontested market space. rather than going head-to-head competition with other companies that provide the same product or service. So, for example, type 2 diabetes drugs, such as these SGLT2 therapies that are all being worked on by different companies, they're going to compete with one another head-to-head. So that will be considered a red ocean market because there's a lot of competition. Typical SWOT analysis, where you take a look at the strengths, the weaknesses, the opportunities, and the threats. And this is pretty straightforward. So the takeaway message here is that really the only threat to Kaleido is the uncertainty of the recently passed Affordable Health Care Act, also known as Obamacare. Just not sure how that's going to impact really essentially everything. And again, the marketing strategy is relatively straightforward. Obviously, the time and effort that went into commercializing Kaleido was huge, and you would expect for a drug like this to be really expensive, which it is. As we mentioned, it was approximately $294,000 a year for therapy. So without directly speaking to Vertex staff, we can reasonably presume that the reimbursement strategy was based on current therapy benchmarks, probably some comparable therapies, which obviously are not many, but they may have looked at similarly high-priced therapies involving orphan drugs, and then they considered the efficacy and overall economic value. Vertex has other CF drugs in their pipeline, and as we mentioned earlier, they're positioned to dominate the CF market entirely. So, again, according to the stock guy, Jim Cramer, as we talked about earlier, Vertex's current market valuation is $18 billion. And in the scheme of things, that's pretty cheap. So don't get us wrong. We're not trying to pump Vertex's stock. We're just passing along Cramer's opinion. Sales, strategy, and the managed markets. The sales strategy for Kaleido appears to be a stepwise process where it builds on itself. So now that Kaleido is approved for use in patients with this particular CF mutation, Vertex plans to use a combination therapy with Kaleido with the experimental potentiator VX809. And they think that second generation potentiators like VX661 and VX983 very well may treat the majority of the cystic fibrosis population. Vertex is probably not anticipating any market shifts that would impact their continued commercialization activities. And so they don't need to differentiate Kaleido because they don't have any current competition, and it doesn't look like they'll have any. The CF community stays current, and the Cystic Fibrosis Foundation is mature, and it's a very well-oiled foundation that gets a lot of information out there. For our final remarks, as our team leader Anna explained, Ivacaftor really was a shot in the dark. Like we've learned in our class, not many medicines make it to market. So when you think that it took just about 12 years to get from a mere compound to a really what could be considered a revolutionary new therapy that could significantly extend the lives of some patients with CF. This is just nothing short of miraculous. So with that, we hope you've enjoyed our presentation. We also hope that you've learned something new. And most especially, thank you so much for taking the time to view our presentation.